Hello, everyone, and welcome to Chapter 19. This is a redo of replacing an older lecture, so it'll also be some new information in it that wasn't in old lectures. As we get started, I always like to start things off with a quote. I chose this one because I want to remind you that your hard work saves lives, and that's a pint of sweat saves a gallon of blood. George S. Patton. As we go through, please remember I will test on every objective. These objectives are listed. These are the things that a student should be able to do and discuss what's happening. Now, as we get started, let's just talk very basic about what blood. This chapter deals with blood. What are the 5 functions of blood? Blood is a fluid connective tissue with 5 major important functions: transporting of materials, regulating pH ions in the ECF, restricting fluid loss, defending against pathogens, and stabilizing body temperature. So let's look at the idea that I transport things like gases, nutrients, hormones, and wastes. For example, from my lungs, I try to get gases from the lungs into the blood, and from the blood to the lungs, and then get those gases to the tissues that need it. I've got to get nutrients from the digestive tract, the liver, and the fat, and move that to where it's got to go, to the tissues that it needs. And I've got to get hormones, for example, from the endocrine glands or the target cells. Now, I didn't mention here that I've got to get waste products from where they're produced out to the kidneys, things like that. We regulate pH. For example, we can remove hydrogen ions to regulate pH. We can secrete hydrogen ions. We can secrete bicarbonate ions, things like that. We remove things from blood or add things to blood to adjust and regulate that. So we can do things like potassium, sodium, and calcium. We can restrict fluid loss. Blood has the ability to coagulate. We have blood clotting that happens as well. We can defend against pathogens. There are white blood cells in the blood and antibodies that help us fight infections. And we stabilize temperature. How do we do that? Well, when I move my muscles, muscles generate heat. The heat is absorbed, transferred to the blood. The blood transfers that to skin and other body tissues to keep the body warm. All right, here's a big thing. So to get started, this will be the first kind of really, really big thing. And there are a couple things here that are a little different, and that's going to be the numbers that I'll cover later on. And I'll try to keep this drawing for kind of recreate a little of that as we go along. I wanted it all in one slide. It's just you can't fit it all. I may go and try to do some things to this and just I think I know what I want to do to it just to get rid of it. But let's talk about overall whole blood's composition. So let's discuss the composition of whole blood. Okay, the composition of whole blood. How is whole blood? How is it composed? What's in it? Things like that. Okay, so let's start off by really looking at a tube. And let's fill this tube full of blood. Let's put some blood in this tube. We got a test tube. We're going to put some blood in it. Now, since I don't have all the colors I'd like to have here with me, I only have the 3 primary colors and then black. What I want to do is really kind of discuss some things here. All right. And we'll use blue for the other one. All right. Now, what we're going to see is plasma. And we're going to see what is called formed elements. All right. Now, these are the 2 major components of overall blood: formed elements and plasma. Now, when we take a look at this, we're going to see that plasma is 55% of overall blood volume, and it ranges between 46 to 63%. So let's go ahead here and talk about the idea that it is about 55%. So about 55% here that we have is plasma. The remaining 45% formed elements are about 45%. Okay. Now that gets us to the 100%. Now, as we talk about this, what we want to do is discuss each and every kind of thing that we have here. So starting off, let's discuss the plasma composition. Plasma is consisting mostly of water, 92%. And I'm going to actually do that first. About 92% is water. So if I were to look at this plasma, I'm going to see there is H2O water, and that is about 92% of the volume of plasma. Now, plasma is 55% of total blood volume. Of plasma, water is 92% of that, 55%. Then there are plasma proteins, and the plasma proteins are about 7%. Now, that'll be a big area that we'll talk about in a little bit, and I will come back to that, is at 7%. And then lastly, what we're going to have is solutes. And we'll talk about what's in there, okay? Now, as we discuss plasma, the solutes are about 1% of overall. As you can see, solutes usually exist as about 1% of overall blood volume, about 1%. Let me put that in my drawing. I forgot to do that. I keep hearing my dogs upstairs and it gets me kind of distracted. Okay, about 1%. Okay, so let's take a look at that. So now what I want to do, guys, is talk about the formed elements, and that's 45%. Now, formed elements mostly are going to be made up of red blood cells. Now, what I'm going to go ahead and do is talk about those of what the formed elements actually are. We're going to see mostly red blood cells, R, B, C's. Then we have this thin little area here that I've got. If we were to look at it, we would see it kind of like that, which is actually referred to as a buffy coat. And that is made up of white blood cells, WBCs, and platelets. White blood cells and platelets. Now, this buffy coat, these white blood cells and platelets, what we're going to notice about them is you have about the platelets, which aren't on here till now. Together, these only, the platelets and white blood cells combined are about 0.1% combined. So this is about 0.1%, 0.1% combined, which makes your white blood cell, your red blood cells, about 99.99% of overall formed elements. Now, we'll get into these numbers and talk about the numbers of these momentarily, okay? So that is your overall major composition. Now let's look a little bit more in detail. What I want to look in more in detail is that you're talking about how did we get the blood that we have here? How did we get this? How was this obtained? Well, there are two methods to obtain blood. One is called a venipuncture. Venipuncture is taking a needle and putting it into a vein, usually a median cubital vein, sometimes using a median antibrachial. And using these, but median cubital is the most common. Why do we do that? Veins have thinner walls, which are lower pressure, and they heal better. It's used for a metabolic panel. It has a lower oxygen, higher CO2 content and also less nutrients than arterial blood has. Arterial puncture or arterial stick is used usually at a radial artery or the brachial artery at the elbow. So we go to the wrist at the radial artery, or we go to the ulnar artery, or rather the brachial artery at the elbow, and it's used to measure gas efficiency of gas exchange at the lungs because of the higher oxygen content and lower CO2 content, higher nutrient count in the arterial blood. All right, let's talk some properties. Blood has some major properties. Number one, it's a little bit warmer than average body temperature. Average body temperature is 98.6 degrees Fahrenheit, 37 degrees Celsius, so a degree Celsius greater than average body temperature, slightly higher than a normal body temperature. Blood is 5 times thicker or 5 times more viscous than water. So, in fact, blood is thicker than water. I know that I was telling my class that phrase actually comes from that when people say blood is thicker than water, it's actually the blood of the covenant is thicker than the water of the womb. Not talking about family. The pH is about 7.35, 7.45, average is 7.4. Males have about 5 to 6 liters, where females have about 4 to 5 liters. We can say 5 liters is kind of in the middle realm there. So we could say about 5 liters, kind of fits both. Both can have, that's like low end of males, high end of females. So about 5 liters. So 5 to 6 liters for a male, 4 to 5 liters for a female of blood volume. Now, plasma is actually, as we said, 55%. It can range from 46 to 63%. It's pale yellow in color. It and the interstitial fluid are major extracellular fluid components. In it, now what I want to do is talk about their proteins. So we're going to talk about the 2 important groups and then 2 more. So there's 4 of them I want to talk about. So there's 4 of them I want to add here with plasma proteins. And I'm going to use this pen 1, 2, 3, and 4. I'm sorry if it's I might move solute down a little bit. Trying to make room. That's the trouble with not doing this on an actual board. Is a big board at work. When I'm teaching, I have plenty of room. But the plasma proteins are definitely going to be very important. Okay, so let's talk about them. Okay, I'm going to use the finer tip pen. So as we go through them, we're going to list them. We're going to start with albumin, which is 60%. It is by far the most abundant. It has a lot of functions besides giving what is called the osmotic pressure of blood, helping maintain the osmotic pressure of blood. It's a major transport protein for lipids, steroids, things like that, things that are hydrophobic. And it's 60%. It's called albumin. Albumin will be the most abundant, so we'll say albumin. Albumin is about 60% of overall plasma proteins. Then we have our globulins. Now, our globulins is a big factor here. I will talk about globulins. The globulins, as we can see here, they are about 35%. Now, we'll talk about the examples of globulins. Globulins come in a variety of different types. They're the antibodies, immunoglobulins, they're called, IGs. They're transport globulins like transthyretin to transport some of the thyroid hormones, transferrin to transport iron, and apolipoproteins to transport triglycerides and other lipids. Then we need to get fibrinogen. Fibrinogen. Now, fibrinogen is 4%. Fibrinogen. Now, fibrinogen. When you see the suffix gen, what I want you to think of is it's got to be activated. Okay, so we said it's 4%. Now, lastly, what I want to talk about, guys, is the regulatory proteins. And these are just called regulatory proteins. These are less than a percent. They're less than a percent. They're a very small number. They are less than 1%. They are, now, something I want to mention about, and I thought about doing this, is most of there are peptide hormones, there's other liver proteins. Actually, 90% of all proteins in plasma come from the liver. Antibodies, peptide hormones, things like that. All right. Now, one of the things I also want to talk about is the solutes. Solutes are things like electrolytes, organic nutrients, and organic wastes. So there are electrolytes, organic nutrients, and organic wastes. So we have electrolytes, we have nutrients, and we have wastes. Now, we talked about solute a little bit, and solutes, when I discuss the solutes, I don't spend a lot of time talking about the solute concentration and how many. When I actually get into the solutes, we don't talk about the other solutes and their major concentrations. That will be done in fluid electrolyte acid-base balance. Now, red blood cells are 99.99%, where together platelets and white blood cells form about 0.1%. And red blood cells are by far the most abundant. Now, something I want to do here is if you take a microliter, you will actually find red blood cells 4.2 to 6.3 million. So there's about 4.2 to 6.3 million. 4.2 to 6.3 million in 1 microliter. Okay. Now, I will try to give these numbers. Let me go ahead and discuss some of the numbers that I've got so I can put them in the drawing. I also want to go ahead. I know it's kind of me breaking the script a little bit, but I want to do it on purpose. This is kind of purposeful. White blood cells, we'll get a number there. They're about 5,000 to 10,000 white blood cells, so WBCs, 5,000. So if we take about the WBCs, just WBCs here, so if I have WBCs that I have, remember, they are WBCs. They are sitting there at about 5 to 10,000. Per microliter. We'll talk about platelets as well. And what I was going to do is I wish I had this on one slide, but I don't. And it's kind of annoying to me that I don't have it that way. That's something else I want to do at some point and talk about platelets, talk about the thrombocytes, and I must not have put them in there yet. I must have left that number out. And that's my fault for leaving that number out. I will try to fix that and remedy that in another recording. All right. So we've gotten through all the components of major bit of blood. And we could say, guys, about 45% of whole blood there. What I want to do is discuss a little bit more about blood, and I'm going to need to talk about hematopoiesis a little bit. And as we talk about hematopoiesis, what I want to help you understand about hematopoiesis is this is red blood cells and other blood cells, white blood cells, platelets being formed. This is the production of blood cells. Hematology, study of blood, hematoblood. Poiesis formation. This is the formation of blood's formed elements. Formed elements of blood are being formed here, okay, in a process called hematopoiesis. Now, in hematopoiesis, What we want to talk about with hematopoiesis is basically this. In hematopoiesis, what you do is in hematopoiesis, you have a cell sitting up in the red bone marrow called a hemocytoblast. A hemocytoblasts. Now, hemocytoblasts are kind of like a college freshman. And when you walk in as a college freshman, one of the things you do is you try to pick a major. Now, you might have a couple majors in mind. Maybe you decide you want to be a nursing student or you might want to be a social worker. You know you want to help people. So you say, I either want to be a social worker or I want to be a nurse. So you get to pick one of two majors. Now, there's one kind of thing called a myeloid stem cell and a lymphoid stem cell. Now, what are these cells? Myeloid stem cells, they're going to become the red blood cells, RBCs. They're going to become most white blood cells, WBCs. And they're going to become the platelets. So what's left? Lymphoid stem cell becomes one kind of cell, lymphocyte. Lymphocytes. That will be, for example, is B cells, T cells, natural killer cells, in case you guys didn't know. Now, that is basically myeloid stem cells become all blood cells except lymphocytes. Okay? All righty. Now, what happens is these cells, very early on, embryonic, about third week, they're embryonic blood cells. They appear. They come from the mother's yolk sac. They show up. She made a yolk sac from the egg. So at birth, the process starts happening. They go to what becomes the liver, spleen, thymus, and bone marrow, where cells are going to be produced and grow up and mature. And they make blood cells. The stem cells in red bone marrow, to right now, it's the only site we do this after birth. There is some evidence to suggest this may not be the case. I've read some papers that have suggested they have found actively dividing cells for blood stem cells in the lung. Many white blood cells are also made in the bone as well. Now, these stem cells, what they do is they become what's called hemocytoblasts. Hemocytoblast picks a major and might decide it wants to become a nursing student or a social worker. Okay, sorry, I couldn't get the word to come out. And the nursing student, you go one thing. Now, here's the deal. After you go far enough down the line, there's no really going back. Now, there is, in reality, I can decide I can change my major, and I just know that I may have to stay an extra year or two in college if I change my major. But cells can't do that. Cells, once they go down the path, they forever are on that path. Myeloid stem cells will produce red blood cells and most white blood cells except for lymphocytes and platelets. They make all blood cells except for lymphocytes, where lymphoid stem cells give rise to the lymphocytes. Now let's talk a little bit about how we produce this. Now, in the bone, there is what is called the hemocytoblasts, and they are in the red bone marrow. And in the red bone marrow, what I want to help you guys understand with red bone marrow is this. Red bone marrow, being the site of production, is got to know that we have low red blood cell count or low oxygen. So how do we do this? Well, red blood cell production is stimulated by low oxygen levels or low red blood cell count, anemia or hypoxia. Now, to determine this, there are chemoreceptors that are located in the kidney and liver, and they detect the oxygen deficit. And they release from the kidneys anyway, releases erythropoietin, EPO, and erythropoietin, EPO, will stimulate red blood cell production in the bone marrow. The production will turn on. That will start to get the red blood cell numbers up, which will increase oxygen saturation, which will, as the oxygen levels go up, EPO turns off. Now, something interesting about red blood cells, when they're made, they lose their nucleus and they do what is called an anucleation. They anucleate. And they not only lack a nucleus, they lack mitochondria and ribosomes. So they only can make ATP through glycolysis. Now, this matters a lot. If you ever have to learn oxygen hemoglobin dissociation curve, it's important that you know red blood cells can only make ATP through glycolysis, because glycolysis makes a molecule called 1,3-BPG, 1,3-bisphosphoglycerate, which acts as a modulator for hemoglobin and affects hemoglobin saturation with oxygen. They can't divide, they can't synthesize, they are on their own. Now, they live about 120 days. And let's kind of talk about what happens. Is that I've got a red blood cell here. And let's talk a little bit about red blood cells. Let's say I've got a red blood cell. Here's an RBC. Here I have an RBC. Now, this RBC, what it's going to have in it is a bunch of this protein called hemoglobin in it. So it's going to have a bunch of hemoglobins. And we're going to talk about this hemoglobin in a little bit. Now, this hemoglobin, it contains something called heme and something called globin, the globular protein. Combined, this is called hemoglobin. Now, what's going to happen is the liver and places like that are going to get this hemoglobin. These red blood cells, after about 120 days, let's say about 120 days, liver, spleen, bone marrow. What these guys all have is they have macrophages. And what these macrophages are going to do is they're going to get this, they're going to pop it open, and what we're going to get is we're going to release all this hemoglobin. Okay, we're going to release this hemoglobin. So now what we want to do is zoom in on the hemoglobin that we've released. We're going to release all this hemoglobin. Okay, now all this hemoglobin, we said there's globin and there's heme. Heme is like a disc. And heme has an ion, Fe2+, iron 2+, Fe2+, ferrous iron, Fe2+. Now, basically what's going to have to happen is we're going to take this globin here, this hemoglobin, and we're going to separate this. We're going to take the globin and we're going to recycle it. But we're worried about is the heme. We're worried about this heme with the iron 2+, Fe++, Fe2+. What we're going to do is we're going to release that Fe2+, this heme, okay, our heme, is going to get released. And this heme is a pigment. And what's going to happen is we're going to take this heme and we're going to break it down and we're going to make a substance out of it called biliverdin, which is the green pigment found in things like BIOL, things like that. Then we're going to convert it to bilirubin, which is more yellow, And this is what happens in jaundice. And this is why you will see this stuff here. Now, eventually, these things can go out urine or feces. And anytime the color of urine and feces changes, could indicate that there is a bleeding disorder. Okay, that you have a bleeding disorder. We get rid of it basically through urine and feces. So, especially now, bilirubin, it becomes what's called urobilin and makes urine yellow. This is why urine is yellow. All right. Now, a typical cell, red blood cell, lives. Now, the thing I forgot about is to show this, and I forgot about it. This iron gets released, and there's a protein that grabs it up called transferrin. Think Fe equals Fe plus plus is ferrous. Ferrous. Okay. So transferrin is going to pick it up, and I can take it to where we make more red blood cells. Okay, I almost forgot to talk about that. I almost forgot to mention it. So the heme is the pigment. The globin is the protein. The iron gets released in the heme. It gets reused and stored by going on a transferrin, transported back to like developing red blood cells. The heme is then turned into biliverdin. Biliverdin is green. Biliverdin gets turned into bilirubin. That's yellow or orange. That's what jaundice patients have. And these get excreted through like bile, urine, and feces. Urine and feces clinically are the big ones I wanted to draw out because those are the ones that can indicate blood diseases if your urine or feces colors change. And this shows you guys what's going on there. Also what I drew out. Okay. Now, what's going to happen is, guys, in order to make red blood cells, we need vitamin B12 for blood cell growth. We need iron from our diet so we can make hemoglobin. Hemoglobin is a very important protein. Now, I'm going to talk about hemoglobin more in detail in a little bit, but red blood cells are basically bags of hemoglobin. And these bags of hemoglobin, for example, you have about, in a overall human, has about 25 trillion red blood cells. And there's about 25 trillion red blood cells, and each red blood cell has about 250 million hemoglobins each. So that's 250 million times 25 trillion. That's a lot of hemoglobins. Okay? That's a heck of a lot of hemoglobin. Now, what I want to help you guys understand, basically hemoglobin is what is the pigment that binds to oxygen and carbon dioxide, gives blood its red color. A single red blood cell, like I said, has about 250 million hemoglobins. That you can have about 1 billion O2 molecules in every single red blood cell. Now, a single drop of blood can have over 1 million red blood cells. They live about 120 days. Now, let's talk about what is hematocrit. Hematocrit is a percentage of whole blood composed of RBCs. Now, to discuss hematocrit, what I want to do is talk about with hematocrit is a percentage. Now, hematocrit, let's say I've got that test tube. Now, let's say I go in, guys, and I take my blood. I put some blood in that tube. I add blood to this tube, and we fill this with blood. And let's say we said about 45% roughly is formed elements. And of that 45%, remember, some of that is not red blood cells, okay? Only some of this is RBCs of that 45%. Now, let's say we have that, and this is my red blood cells, okay? The rest of this is plasma, Okay, the rest of this is plasma. So there is this value right here. This percent is called hematocrit. Hematocrit, or sometimes called packed cell volume. Why? What they do is they put blood in a tube, centrifuge it, pack it down by heaviest cells at the bottom, lighter cells and fluid at the top, and you have what is called a packed cell volume, or hematocrit. This percentage of whole blood that is composed of red blood cells is the hematocrit. So if we were to find out that 45% in this case of the blood was red blood cells, they would have a hematocrit of 45%. Now, this can help, for example, if hematocrit is low, we would say you're anemic, for example, okay? So, what we'll see, guys, I'll talk about hematocrit a little bit more. Hematocrit is usually about 42 to 54% for a male and 38 to 46% for a female. And this is due to men have higher levels of androgens. And then the thin buffy coat that are made up of white blood cells and platelets. So that is called your your hematocrit or packed cell volume. Now, something I want to also help you guys understand about this. Hematocrit's a very important laboratory value, so you got to know what that hematocrit is. Now, do not worry about the normal hematocrit. I will not test you guys on what a normal hematocrit is. You should generally know that a normal hematocrit, based on the percentages of red blood cells I gave you. Now, one thing I can tell you guys about red blood cells, about RBC structure, is now there's loads of things we can talk about, but a red blood cell is what we call biconcave. It is skinny in the middle and wider on the edges. This increases surface area. Okay. They have a biconcave disc. Now that does matter. I talk about they have no nucleus, no mitochondria. They make ATP through glycolysis, which is an anaerobic thing. This helps give it increased surface area where it's thick on the edges and thin in the middle, like pizza. It's the increased surface area for gas exchange. They can fold up and stack. They can stack up in what's called a rollout. And then inside there's a volume. If we were to go in and figure out what is the volume inside, if we were to go in here and basically measure the space inside this and measured it and said how much, and we did it for a bunch of red blood cells and averaged it, we call this average volume of red blood cell the mean, which is average, corpuscular volume. Sorry if you can hear my dog bark. Mean corpuscular volume. If you had a bunch of red blood cells and we found what the volume of that cell was and averaged it together, we'd have mean corpuscular volume, the average volume of red blood cell. Don't worry about it. Now, we talked a little bit about hemoglobin, and I want to help you guys understand hemoglobin. Hemoglobin is a very important protein in blood. Without hemoglobin, we could not carry enough oxygen to be able to keep a body going. Now, sorry if you can hear my dog bark. Too far along to stop. Now, let's talk a little bit about hemoglobin. Now, hemoglobin is made up I don't know if you can actually hear her. I can't tell, and I apologize if you can. She is very loud, and normally, all right. So hemoglobin. Now remember, we have what is called heme. Remember, we also have what is called, sorry, globin. My brain, my dog's barking. It's got me very distracted, and I can't think when I'm hearing her. Okay, so we have our heme. I apologize. And the heme has Fe2 plus, Fe plus plus, lost 2 electrons. All right. And this makes hemoglobin. Now, hemoglobin, what it does is you have one hemoglobin. We have what is called an alpha hemoglobin and a beta hemoglobin. And then we can have, for example, alpha and beta globin. And what you have is 2 alphas, 2 beta hemoglobins. Okay? So there's actually 2 alpha chains and 2 beta chains. Alpha, beta, just like I drew, okay? Now, it has 4 total changes of quaternary protein. And here you can actually see the heme. Heme is a disc, and basically remember a red blood cell is just a bag full of hemoglobin. These hemoglobins have 4 chains with a heme in the middle, Fe++. And what the heme will do is it's the thing that binds to the oxygen. So, for example, the oxygen will bind to this, and you can actually get 4 O2s. Okay. Now, what happens is you got what is called oxyhemoglobin and deoxyhemoglobin. Now, I oftentimes like to show hemoglobin. There is what is called HB, which is deoxyhemoglobin. And then we add O2 and we make HbO2 or oxyhemoglobin. Now, these 2 globins, oxyhemoglobin and beta hemoglobin, you have oxyhemoglobin is bound to oxygen. It gets more red, it gets a bright red. Fetal hemoglobin has even higher affinity for oxygen than does adult hemoglobin. When oxygen releases, we call it deoxyhemoglobin. When carbon dioxide is bound to it, it's called carbaminohemoglobin. All right. Now, these are not on the test. I want to let you guys know. In infants, there's about 14 to 20 grams of hemoglobin in 100 mils of blood versus less. You notice that in adults, there are less. Actually, growing babies per 100 mils have more hemoglobin, and the hemoglobin has greater affinity in an infant. It's about 13 to 18 for a male, 12 to 16 for a female. Again, females are smaller and have lower numbers of blood cells. So here's your hemoglobin. You can see 4 different subunits. In the middle, there is a heme. Heme has an iron in the center. Those blue balls are the nitrogens. All right. Now, bruising. We're going to get into the really hard stuff in just a minute, but bruising is something that is happening when blood cells are damaged, and they release the breakdown of biliverdin, bilirubin, things like that happen outside in the tissues. This creates the colors of bruising, is the hemoglobin breakdown happening in tissues that enters the bloodstream, and you get yellow, green, blue colorations to a bruise. And that's because the colors of bruising are to damaged RBCs release hemoglobin in the tissues. Now, let's see if you guys can think through this. There's an autoimmune disease called hemolytic anemia, the condition where antibodies destroy red blood cells faster than the body can produce them. Jaundice, weakness and lethargy, and dark urine. Now, why is that? Well, we have to understand first. Think about this. Go ahead, think about it, pause, and then I'll give you the answer. Okay, I hope you paused. If you didn't, pause. But here's the answer. Jaundice, guys, remember bilirubin accumulates when blood cells, after they pop, we recycle the hemoglobin. Weakness and lethargy. You're destroying them faster than you can make them, so you're not getting oxygen. You can't make ATP because you need oxygen to make ATP. Dark urine, you're getting rid of all those through urine. That is where we get rid of a lot of the chemicals from blood. As I said, coloration to urine change can show blood disease. All right. Hope that helps. Now let's talk about blood type. This is the hard thing, blood type. Now, blood type, it can make a lot of sense, and if you let me explain it, then it's going to make sense. You guys will understand it. Now, to do this, we need to get a few definitions down. The first definition is the word antigen. What is an antigen? An antigen is a molecule that can cause an immune response if antibodies are present. They are things that can result in immune responses. Antigens cause immune responses when there are antibodies against them and they're around. Usually an antigen is something on the foreign cells. But we could say our own antigens as well. Antigens are the molecules on the surface of cells. Antibodies are immune system things that we produce to attack any antigen it encounters. Our bodies produce antibodies. Antibodies, when they attack, cause a clumping called agglutination, which will lead to hemolysis. Your blood type is determined by your genetics, which is the absence or presence of certain antigens on your red blood cells. Now, the antigens can be A, they can be B, or they can be RH. Those are the most important. There are others, but those are the most important. Now, let's talk about this. Let me help you guys understand this. This is where we're going to get really, there's a lot to talk about, and I want to help you understand it. So let me help you understand it. We have type A blood. We have type B blood, we have type AB blood, and we have type O blood. Okay, these are blood types. Now, we're just dealing only right now with AB blood type system, not the RH yet. I'll talk about RH right now. We're going to deal with AB blood type system. So here we have RBCs. Here's an RBC, okay? These are all RBCs. Now, what we have on RBCs are antigens. Now, let's say A. A has a very specific antigen, and I'm going to draw it like that, and we're going to call this the A antigen, okay? The A antigen. So let's say this is an A antigen. Okay. Now, in the fluids, there are antibodies that are anti-B antibodies. The antibodies are anti-B. Now, some of you may call me out and say, Ledford, you guy at Myelination, if you're not one of my students and you're just one of my followers, you might be saying the blood type antibodies aren't shaped like that. They're pentamers. I know, but I'm just being simple. All right. B. Let's say this is the B antigen. Has only B antigens. But the antibodies it has will be anti-A antibodies. Now, guys, let's think through this. The last thing you want in your plasma is antibodies against your own blood type. That would kill your own cells. So you never have antibodies against your own blood type. Now let's think about this. How do you get AB? AB has the A antigen and it has the B antigen both. It has both A and B antigens on it. So the plasma will have no antibodies. And this is called the universal plasma donor. Universal plasma donor. Now over here we have O. There are no antigens. So they will have antibodies that are both anti-A and B. Anti-A and anti-B antibodies. They will have both anti-A and anti-B. There is nothing on it to attack, so it has both. Now, let me kind of explain to you how this happens. I'm going to talk about basically what happens, guys. Genetics determines what antigens go on the red blood cell surface. Now, as you eat food, as you grow up, you start to eat food as you get as you're a baby, you start to eat solid foods, and on the foods that you eat are also antigens. Now, when you eat food, let's say the same molecule for the B antigen is on the food that you eat, and you have type B blood, your body will ignore it. But if you have type B blood and you eat food that has A antigen on it, your body will start to make antibodies against A, ignoring any of the B antigens. As you eat foods and consume that, your body will start to make antibodies against the blood type you don't have, the other one, like the antigens you don't have. So you only make antibodies for antigens you don't have. That's how come you get them. Don't worry about how you got them. It's not in the notes, but I always teach it. Type A, which is about 40% of people roughly, have A antigen. These RBCs have type A blood. This is surface antigen A, anti-B antibodies. The antibodies are very small in concentration. The type B, which is red blood cells, have the B antigen anti-A antibodies. AB, which is actually both A and B, have no antibodies in the serum or in the plasma. And type O is actually no surface antigens, but they have anti-B and anti-A antibodies. So they have both in the plasma, which means, but they're very small amount. And I always want you guys to know they're very small amount. Now here they use circle for A and triangle for B. It doesn't matter what shape you use. I'm just using it as an indicator to give you guys an idea. Now, the next thing is called the RH factor. Now, RH factors, what happened is this is a whole different ball game. RH factors are, by the way, A and B are what we call codominant alleles. If you guys had a good high school biology teacher, you may have learned about codominant alleles. Or if you took General Biology 1, you learned that some alleles are codominant. And codominance means that an allele, for example, will be both have like be equally dominant to another allele. So by having both alleles, you have both phenotypes. So now here, this is completely dominant. You either are Rh positive or Rh negative. You're not Rh middle. There are multiple Rh antigens. The most common is called D. Rh positive, or just positive, like A positive, B positive, AB positive, O positive, is that you have the Rh factor. The Rh negative, or just negative, is like A negative, B negative, AB negative, O negative, means you don't have the Rh. Now, the thing about this is the only way you get antibodies against these is exposure to the antigen. You have to be Rh negative and be having been exposed to Rh positive blood once. Then, so you're a Rh negative person who gets exposed to Rh positive blood, then Rh positive, Rh negative people who have seen Rh positive blood are said to be sensitized, and then sensitized people will make anti-Rh antibodies. And this can kill you. The wrong blood type can kill, and Rh incompatibilities are very serious, but also can kill somebody when you give. Back in the day, what they did is on battlefields, they would basically put a needle with a hose from one person's vein into a needle with a hose on another person's vein and the same hose, and they would drain blood from one person to another person. Sometimes the person who got the blood died, and sometimes they lived and they didn't know why. What we found out is there are the antigens on the surface, and sometimes these antigens from the donor's blood is incompatible with the antibodies in the recipient's blood. So O negative is called the universal donor because the RBCs have no antigens. So the donor's blood goes into your recipient blood, and the recipient's antibodies have nothing to attack. Whereas AB positive is called the universal acceptor because they have no plasma antibodies. There is no, they have no antibodies to attack the donor's antigens. And because there's no anti-A, anti-B antibodies in AB positive, we call that universal plasma donor. Now, what happens, and I'll talk about these, and I'll go through it and talk about it, is agglutinogens. These are your antigens. These are your own blood type. If you're type A, your agglutinogen is A. If you're type B, your agglutinogen is B. If you're AB or O, okay? Those are your agglutinogens, your own blood type. The antigens ignored. The agglutinins, those are the antibodies. Your antibodies are called agglutinins. Agglutination is when an antibody finds an incompatible blood type and that it leads to hemolysis and kills it and can actually cause death. This agglutination can cause strokes, heart attacks, hemolysis killing it, causing jaundice. So a compatible donor is and recipient. The donor is called compatible if they are compatible. The donor and the recipient's blood don't cross-react. That's important. Your agglutinogens, though, are so diffuse when we put them into a patient that it is your agglutinins, your antibodies, are so diffuse and diluted in a pint of blood. Usually they have no impact. So, guys, with that statement, keep this mantra. It is donor antigens versus recipient's antibodies. Donors antigens versus recipient antibodies. Okay? If you have that down, it's going to help you guys so much understand this. Okay? So let's get into it. What I'm going to do is draw this out, and let's say I've got 4 red blood cells. We have A, we have B, we have AB, and we have O. Now, you got to remember the antibodies, but let's remember A. I'm going to use A has the A antigen, B has the B antigen, AB has both, and O has none. Remember the antibodies, though. So let's take a look at this. Let's see who can A. First thing, you can always give to yourself. So A can donate blood to A. Absolutely. But can A give to B? Let's think. A has A antigen. B's plasma has anti-A antibodies. So A cannot give to B. Because remember that B blood has the anti-A antibodies. Can A give to AB? AB has no antibodies. So A can give to AB. can A give to O? O has both anti-A and anti-B antibodies, so A cannot give. So A can give to itself, and A can give to AB. B can give to itself. B can give to itself. B, can it give to A? A has anti-B antibodies in its plasma, so no. B, can it give to AB? Yes. AB has no antibodies to attack the B antigens. Can B give to O? O has both anti-A and anti-B antibodies, so the answer is no. It will attack it. Let's move to AB. AB can give to itself, but can AB give to A? A has anti-B antibodies, and AB blood has B antigens, so no. Can AB give to B? B has anti-A antibodies, and AB has A antigens, so no. And AB, can it give to O? No. O has both anti-A and anti-B, and this has A and B both on it, so it's double trouble. So AB can only give to itself. Now O has nothing on it. So can O give to itself? Yes, safe. Can O give to A? Yes, O can give to A because there's no antigens for the anti-B antibodies to attack. Can O give to B? Yes, there's no antigens for the anti-A antibodies to attack. Can O give to AB? Yes. There are, again, no antigens for the anti-A and anti-B. Sorry I got interrupted there, so I hope that I haven't missed anything. So this is why we call O universal donor and AB universal recipient. You will need to be able to understand this. So this is definitely going to be where a higher order kind of question comes from on this test. So definitely understand this concept. And I hope it takes a little time. You got to remember it's donor antigens versus recipient antibodies. And here you can see this. For example, I have plates that I have anti-A antibodies, anti-B antibodies, and anti-D. That's the RH. So if I put blood and I get clumping in anti-A, no clumping in anti-B, and clumping in anti-D, then this is A positive blood. No clumping in A, clumping in B, and clumping in D, this is B positive. Clumping in anti-A, anti-B, and anti-D is AB positive, and no clumping is what is called O negative. Okay. And here I've done this where I just basically give you a table that says a person with A can receive blood from A or O, B can get from B or O, AB can get A, B, AB, or O, and O can only get from itself. O can only donate to itself. And here is an actual full-on compatibility table, including the RH factors. Okay. O negative is the true universal donor. O negative is like the greatest blood type. Okay, that's the best. Recipient, AB positive can receive from anybody, and O negative can give to anybody. That's like your O negative is like the best blood. All right, and I do want to make a point here. This won't be on the exam. And that is that there are distributions of blood type amongst humans, amongst ethnic groups. Now, I will hear people sometimes, they will say on the news, they say, we need more African American individuals in the community to donate blood, or we need more Latino American, Filipino American, African American, we need more Latin American individuals to donate blood. Why that is these different groups have different percentages of blood types that are more common in those groups. So we want those so we can actually get those more common. That's actually the reason why they do it, is it helps us get a higher number of needed blood types. Don't worry about it. I don't cover too much in detail about that, but I just want everybody to know it. If you can put an X for all the incompatible crosses, which right here, these are checks and X's. If it's got an X, put a check for compatible and an X for incompatible. Right, there's your answer. Okay. All right. One of the last things we want to talk about today is erythroblastosis fetalis. I will test on it. I don't know if everybody does, but I do. A few other people do, but I test on it. It's erythroblastosis fetalis. Now, remember, the only way you can make anti-RH antibodies is you have to be RH negative. You have to have been exposed to Rh positive before, so now you're sensitized, and then you get it a second time and you make anti-Rh antibodies. So what happens is, let's say I have a mother who's Rh negative. She has a child who's Rh positive and she gives birth to it. What happens is, upon birth, the placenta detaches and hemorrhages, and the fetal and maternal blood mix. And what happens is mama will develop anti-Rh antibodies. Now, this is fine because there's no baby in her to be attacked. Now, the thing is, this comes down to the idea that antibodies can cross placenta. Now, if she has a second baby who's Rh positive and she's Rh negative, her blood type doesn't change, the antibodies that she makes at Rh positive will go in and attack. She makes anti-Rh positive antibodies, anti-Rh antibodies in attack, and it can actually kill the fetus. It usually dies after birth, called hemolytic disease of newborn. And right here you can see it. The first pregnancy, she is Rh negative. The baby is Rh positive. When the fetus hemorrhages at delivery, not the fetus, the placenta hemorrhages at delivery, the blood mixes, the fetal blood mixes, and she makes anti-Rh antibodies. And then she gets pregnant, and the anti-Rh antibodies cross placenta and attacks the fetal blood supply and fetal tissues and can actually kill it, killing her second child. It is a serious thing, so we use a shot called RhoGAM. Which RH, RhoGAM. All right. Let's see if you can do this, and this is the last thing. Guys, so let me go over it. I want you to try to answer it in your head. So when I'm done, pause it, and I'm going to urge you to pause, see if you can answer it, unpause it, and see if you got it right. So let's say you guys got a job working in a lab, and you're testing a patient who's going to surgery. You take a tube. Tube 1 has anti-A antibodies in it. Tube 3 has anti-B antibodies in it. And tube 2 has anti-B. Tube 3 has anti-RH. So there's anti-A, anti-B, anti-RH, or D. 1, 2, 3. You add a drop of the patient's blood to each tube. Tube 1 agglutinates. Tube 2 agglutinates. There's no agglutination in tube 3. What is the patient's blood type? Pause, and then come back, see if you got the answer right. All right, pause. Now, don't. Okay, I hope you paused. So here, if we have anti-A in the first tube and it agglutinates, they obviously have the A antigen. Anti-B in the second tube, it agglutinates, they have the B antigen. So we already know they're AB. But there's no agglutination for RH. So this person is AB negative. All this is right here is this. But I chose AB negative because I didn't do AB negative. All right. Guys, this concludes the first lecture. I hope you found it useful. I hope you guys enjoyed it. I hope you learned. We'll see you in the next one, starting with white blood cells. Guys, thank you very much, and we'll see you in the next one.